All right, this is chapter 27 on prokaryotes. Basically, we're looking at all the various details and types of prokaryotes that you might encounter. There's thousands, millions, even just in a single handful of soil. We actually do some of that in Quest, where we take some soil samples and look at bacteria and so on. So they're all over the place, even though they're tiny, including in places that are very acidic or cold or even very hot, thermal springs and so on, like you can see in that picture. So there's all different kinds of genetic diversity. A lot of what we're learning with the diversity is through molecular studies. And structurally what we do know is that they are unicellular. They can form colonies where they connect to each other and exchange nutrients, become a family if you like. But individually they are all unicellular and they can function as such. Now they come in all different kinds of sizes and shapes. Cocci on the left you see are spherical. Rod-shaped we call bacilli or bacillus is singular, and the last one spirili or spirals you can see on the right there. All different kinds of shapes, and that's just only 3 of them. So they all just about have cell walls, which are very important structurally and for protection, helps them to maintain their shape. And should they be placed in a hypotonic environment, which means an environment with lower salt concentrations than their internal environment, they would be predisposed to taking in water and rupturing. Water will go towards the hypertonic environment inside the cell. You haven't learned those yet, but that's okay. So to prevent them from bursting, they have a cell wall, much like the reason why plants don't burst when you water a plant. So we can partially identify bacteria by their cell wall composition. And so we've divided them into 2 groups, again, gram-positive and gram-negative. And you've heard this before. Gram-positive bacteria have a very large amount of peptidoglycan here that you can see. And they actually, the peptidoglycan is on the surface of the cell wall. So when you dye it or stain it with the stain, the violet dye, it gets really well stuck in the peptidoglycan. And when you rinse it out, it makes it purplish. And over here you see the gram-negative bacteria, which is much less, if any, peptidoglycan, and it's also in the middle, so it doesn't hold the dye very well, and when you rinse it off, it turns pink. In general, the gram-negative bacteria are those that are far more harmful to humans. And here you can see an example of the cell wall on the interior, and then the cell wall in this case is covered by a capsule, which is a bunch of glycoproteins, some polysaccharides, those are sugars and or proteins that help to protect it and to adhere to surfaces and so on, as you can see there. Some prokaryotes also have things sticking out of their cell walls, like fimbriae or pili, and this allows them to stick to other individuals and their structures and so on. They're basically just these legs sticking out. They're not used for locomotion. They are only used for adhering. Locomotion is done, or motility is done, by other structures, including these flagella, which in prokaryotes are different from eukaryotic flagella. They still are flagellated, but structurally they're made up and are slightly different. And bacteria have the ability to exhibit taxis, which is a scientific term that means to move toward or away a certain stimulus directly. Not randomly, it directly responds to the actual stimulus and moves towards the stimulus or away, almost in fear, if you like. The main thing that you know about prokaryotes in their difference from eukaryotes is that they lack compartmentalization, or they don't compartmentalize and organize their different metabolic reactions and functions in different locations within membranes. They do have some membranes, however, such as the plasma membrane, where they have the ability to perform some metabolic functions, such as, for example, photosynthesis. They don't have chloroplasts, but they can do photosynthesis, and it would just take place on the plasma membrane itself, as one example. Most prokaryotes have DNA that's housed in the nucleoid region, as we mentioned before, and it's basically a giant chromosome that is circular. Of course, this doesn't look circular because it's all compacted, but it's a ring. It's all connected end to end, whereas eukaryotic chromosomes are linear. And again, it's in the nucleoid region. It's not wrapped in a membrane. There are also some smaller rings of DNA found in a lot of prokaryotes called plasmids, and plasmids are able to be transferred back and forth between bacteria and provide additional functions that we'll learn about later. Prokaryotes divide by binary fission, which is kind of the prokaryotic term for mitosis and cytokinesis, where they split off all the interior cytoplasm and pinch off the cell membrane as well. And it can happen as fast as about 20 minutes and as long as you can pick. Some bacteria, or prokaryotes I should say, also have endospores, where it enables survival. If they are placed into a harsh environment, they release these endospores, which can withstand the harsh environment, and only once environmental conditions become ideal again, they will regrow from that endospore. It promotes survival, and these things can last a very long time. Now, they can exchange DNA and genes from one prokaryote to another to help them adapt to quickly changing environments. And at 27.2, we'll pick this up in part 2.